Hey, hello everyone. Welcome to our continuing webinar series from BioNano. Today I'm delighted to welcome our two speakers, Ali Bashir and Bobby Sebra from the Icahn School of Medicine at Mount Sinai. They'll be presenting today Diploid Genomic Architecture of an Individual Human. Before I introduce our speakers and hand it over to them, I'll provide a quick 5-minute overview of the BioNano technology. So our platform is called the IRIS platform, and our company really revolves around this core concept that you can linearize very long molecules of DNA in nanochannels, and by doing so, actually visualize the genome directly. And so this is the concept that BioNano has committed itself to developing technology we call the IRIS platform that really focuses on using highly parallel nanochannels to derive the true genome architecture by looking at very long molecules. The molecules we look at are hundreds of kb up to megabase long molecules or even longer. And by doing this, we don't require any amplification of the genome, and we can preserve the architecture and generate higher quality genome assemblies and also elucidate structural variation in native state DNA. So the applications that our technology is really focused on is structural variation detection. Because we look at very long stretches of DNA, we can elucidate a broad range of structural variations. It's also ideal for scaffolding in assembly building, validating of references that you may be working on, sizing gaps in references. And again, because it's a single molecule technology with very long molecules, we can start getting at key haplotyping information, another key application for this platform. As of today, BioNano has, and our customers have mapped over 100 unique organisms, ranging from everything from bacteria all the way up to human and complex crops. So it's a broad tool for use in a number of different organisms. Today's webinar will, of course, focus on human data. This is a slide that depicts the product itself. The IRIS system consists of the IRIS instrument, the IRIS chip, consumable, IRIS prep reagent kits, and a software that comes with it. The IRIS instrument is a 3-laser CCD-based imaging system, sophisticated single molecule imager, all run by the touch screen that you see there. The IRIS chip is really where the core technology of BioNano exists. The NanoChannel arrays are on that IRIS chip. And the IRIS prep reagent kits contain the buffers and reagents that you need to label your sample, which I'll describe briefly in the next slide or two. IRIS View is our visualization software that comes free with the instrument and allows you to see the genome maps that you create, allows you to import next generation sequencing data and do comparisons, and you'll see in today's webinar examples of how our colleagues and collaborators have done that. So very briefly, the way you label your samples with the IRIS platform today is using nicking enzymes. And so we do a nick, label, and repair process. A nicking enzyme is a modified restriction enzyme. It recognizes a particular sequence. In our case, we use 7-base sequence nicking enzymes, and they cause a single-stranded nick. Polymerase then comes in and initiates strand displacement, and behind it, it incorporates fluorescent nucleotides. And so the end result here is that you get bright green labels wherever that 7-base sequence occurs throughout the long molecules in your sample. And on the right here, you can see a sample image from the IRIS system where the blue indicates the long molecules and the green are the labels, or where the nick sites are. Here's a video, hopefully this comes through the webinar okay, where we are actually pulling the DNA molecules. By applying a voltage, we can pull the DNA molecules into the nanochannels. They go through a series of pillars where they unwind from their natural coiled state in solution, and then they stack up again at the mouth of the nanochannels where they're fully stretched. So you can see this video of very long molecules. You see a key up here of 75 kb, so you can see these molecules are on average a couple of hundred kb long. And again, here you'll see it looping again. The DNA molecules unwind through these pillars and are delivered into nanochannels where they're fully stretched. That's the core concept behind BioNano, is direct imaging and linearization of molecules. So here, one of the key features of our technology is that you can collect many images over multiple cycles from your sample. And so the idea here is you can turn the voltage on, pull in a sample of molecules from your DNA sample, take a picture, and flush the rest to waste and bring in the next batch. And we can continue to do this iteratively, 20, 26 cycles at a time, and continue to pull from your sample. And this is really what's allowed BioNano to scale our technology to whole human genome and genomes that are even larger. Once we collect those images, our software automatically identifies where the molecules are, extracts those molecules into digitized versions of those molecules, and we show that here. The vertical bars, black bars, represent where the nick sites are, and the blue is the molecule itself. We then feed those molecules into our de novo assembly algorithm. In a nutshell, it's a pairwise comparison of every single 2 pair of molecules in the batch, and we look for overlaps in those nick patterns. And as those overlaps are identified, we simply grow a map into a complete genome map. And we indicate for the rest of this presentation, you'll see these types of structures here at the bottom. This is really what how we visualize a genome map, with the vertical bars being, again, where the nick sites are. And so the end result here is that you can do your science with these genome maps. Whether you're scaffolding or you're doing structural variation, here I show a comparison between our blue genome map with the reference sequence. And oftentimes, whatever we're comparing to will show in green. That could be a reference. It could be next generation sequencing contigs. It could be even another genome map taken on the iris system. So that's the basic fundamental quick overview of the technology and how genome maps are created. You can get a lot more information by contacting us or just simply going to our website at BioNanoGenomics.com, where all the technology is described in more detail. Okay, with that brief introduction, it's time to move on to our webinar for today. And it is my great pleasure to introduce our speakers, Dr. Ali Bashir and Dr. Bobby Sebra. Both are at the Icahn School of Medicine, and both have dedicated a large portion of their professional careers to the study of long-read technologies and implementing those into genome sequencing in a number of different applications. Both Ali and Bobby were at Pacific Bio for several years, and over the last couple of years have moved over to the Icahn Institute at Mount Sinai with Eric Schott and his team. So the way the webinar will work is Bobby will focus on kind of doing an introduction of long reads and how they're utilized in genome sequencing and into clinical applications. Bobby is Director of Technology Development and Assistant Professor there at Icahn Institute. And then he'll hand it over to Ali Bashir, who is also Assistant Professor in the Department of Genetics and Genomic Sciences. Ali's focus really is on computational and experimental methods and developing these for assembling genomes. And so he'll take over from there and describe the pipeline that they've developed and how they've integrated BioNano data with next-gen sequencing data, in particular Pacific Bio's data, for building extremely high-quality assemblies and doing structural variation discovery. So with that, I think we've switched over, and I'm going to hand it over to Bobby.
All right, great. Can you hear me?
Yep, we can hear you.
All right, fantastic. Thanks for the introduction. Like you said, I'm first going to just take you through some of the motivation of why we decided to do a new long-read NA12878 diploid assembly and how we use BioNano and PacBio data, and then the motivation of what we see your technology being utilized for in a clinical domain, and then I'll pass it off to Ali. So the first slide that you can see here on the left, note that in the lime green sections of the chromosomal representation, you can see where there are gap regions in current human genome assemblies. So these are regions that have never been addressed by next-gen sequencing and are kind of unknown. And so it's one way of looking at what's lost in the current representations of published reference assemblies. And on the right, you can see a next-gen derived assembly where, if you look at my pointer, which hopefully shows up, these peaks and valleys just represent coverage across the chromosomes in a clockwise fashion on the Circo plot. But what's important to note is just the regions I'm pointing out as I'm pointing here, showing where those gaps in the assembly occur, and that's mainly due to just capture efficiency for next-gen technology. So it can either be the way that the capture probes are designed in the technology or just the inherent read lengths of the enzymes that are being used. But as most of the audience probably knows, the longest reads that could ever be contributed in those technologies are towards 500 base pairs. So nothing like what you would generate from the genome maps or from PacBio data that we'll show today. So what we're hoping is to address some of those gaps and show you where the improvements are in the assembly. And so the heterochromatic regions shown in these purple boxes are the most difficult to achieve because obviously they're megabases to tens of megabases in size and are very hard to achieve even with these current technologies. But in particular, the ones that I'm showing in the cyan boxes here are on the order of, up towards a megabase. So they could, in theory, be assembled utilizing genome maps and PacBio data, particularly structural variants in the 10,000 base pair to 100,000 base pair range. So what are some of the specific regions in particular, and how does the next-gen data stack there? Just looking at some of the standard ALU sign elements, and even if you looked at line elements, you'll find that with next-gen Illumina data that basically you have nice coverage up until you reach these redundant regions, and you see nice gaps that occur that can be achieved with PacBio and obviously with your genome maps. So those are kind of the low-hanging fruit of improvements in any reference assembly for human. But what's more important to address and more novel are repeats like what we're showing down at the bottom in the dot plot for MUC5B. If you look at this region of the genome against HG19 and chromosome 1, you see in the purple box that there's a highly structured repetitive element that is actually important in presenting information towards better understanding Crohn's disease and ulcerative colitis related to irritable bowel disorders. So using this just as one example of many that we're really interested in achieving with the longer read technologies. So uniquely in any individual, there are thousands of structural variants that are larger than, a few thousand base pairs. This is just a reference from the Snyder lab at Stanford showing you, again, a chromosomal breakdown. And if you look at the nicks on the side of each of these that are color-coded for the type of variant, you'll just see that there are indeed thousands of variants. In this case, now, how those thousands of variants are assorted in any given individual is unique, so it motivates why you would either want to derive custom assays for targeting these domains in the genome or sequencing with long-read technology for whole genome to better address those phenotypes in the genome and the structural variants and resolving them towards a better clinical path forward. So specifically, the panel that you can see here is a list that we had put together of kind of commonly known structurally variable related regions of the genome. So in the purple box are just trinucleotide repeats. In these trinucleotide repeats, perhaps the most well known is in the FMR1 gene related to Fragile X. And so with some of the recent work done by the Hagerman group out at UC Davis, you've seen, for instance, long-read technologies potentially enabling you the ability to better resolve the number of repeats on a single molecule level for better diagnostic value. So understanding the normal repeat versus a pre-mutation versus a full-on mutation definitely enables a physician or clinician to understand better the severity of the pathology at hand. And so the list that you can see there is just showing you the regions of the genome that could benefit from these technologies. And then outside of that box are just other structural variants, which might be anything from a CNV to a pseudogene In particular, I'll highlight the GBA gene. Here, GBA is affiliated with a disease called Gaucher, and Gaucher disease is equivalent to a lysosomal storage disorder, and basically GBA presents itself genuinely as a 6 kb gene in our standard genome, but it has a pseudogene about 16 kilobases downstream that is so highly similar that you cannot resolve the structural variants or even the SNPs between the two loci using just standard next-gen sequencing. And so obviously to get through the 300 or so known variants in that pseudogene relative to the regular gene, we would benefit greatly from longer and longer read-length technologies. So another way of looking at this in a data-driven approach is to take some of our data shown on the y-axis, which are basically just all TRs up to 10,000 base pairs, and plot them against what's predicted by standard references to date. And you can see that there's a compression that occurs for the most part, whereas if they were 1-for-1 in terms of length, you would basically just get this diagonal line, whereas anything that is truly longer in our NA12878 reference relative to HD19 is going to show up above that axis, and anything that's not compressed will show up below. Do keep in mind that this is definitely just a single individual versus the reference, so there are some discrepancies there, but on average you see the compression, and that's not too surprising considering a lot of these references have been derived using shorter read technologies, so it motivates the benefit of pushing for longer read lengths. Do note that in the red circle here, with PacBio data, which is what we use to generate this plot, you can see that the reads started to trail off at about 5,000–6,000 base pairs, and our mean read length in the particular data set that Ali used to generate this plot happened to have a mean of 5,000 base pairs. So there's some sample or data acquisition that is driving the bias against having points in that, but it's also reference-based, and the concept that, for instance, if you were this point that I'm pointing out, say you were a 9,000 base pair expected reference, but you had an expansion for another 10,000 base pairs, then you would need longer read length to even represent where that point would fall. So there's also some bias there. And then the ones that are undercompressed can still be based off the fact that the longer the read gets, the more likely it is to have a QV drop-off locally, which may trim the read. And even that would be hard to present. So there is room for improvement with both technologies there. Now, specifically to the data that we'll be presenting today, I wanted to lay out for you what we did for the collection of the coverage that was implemented in Ali's assembly. Basically, we have 80x coverage of BioNano genome maps with a mean span of almost 300 kb. So those are fantastic for scaffolding of some of the other long-read data that we have and deconvolving the complex structural variants. And then we have 44x coverage of PacBio data, which is a mixture of both the old C3 chemistry and the current P5. What that really just means is it's a mix of data that had a mean read length of about 2,500, and then the newer data, which had a mean read length in the 6,500 range. So you get an average of about 5 kb across this coverage. And there is 200x Illumina data utilized. Ali will explain how that was used for SNV validation and phasing, but the Illumina data was not utilized in the the de novo assembly itself. So keep that in mind. But basically you can see to derive a high-end 50 of about 30 megabases assembly, you actually do need multiple data formats to come to those conclusions. So the PacBio data just really quickly looked like this for those of you that want to do some comparisons. We started way back in 2013, and the mean was about 2,000 base pairs. These days, we're getting reads out towards 40 kb with maybe a mean of about 6,500. So our data set evolved, but 50% of it was taken back with the old chemistry. So in theory, this type of methodology could actually be done better with current tense data on both platforms, I think. So keep that in mind. And then 2 quick examples of where I think we can drive using the genome maps in particular before I hand it off to Ali are one shown here where, keep in mind the top track is the HD19 reference shown in green, which we'll try to stick to that legend throughout the rest of the slides. And that's what was expected by the reference. The genome map then shows this expansion highlighted in this gray color, which shows essentially this region expanding into what looks like 10 evenly spaced sort of tandem repeats. Now, what I wanted to drive by this example is that you can derive different haplotypes, but also look at the structural variants and potentially use this as a way to count repeats, for instance, for some of the diseases that I had listed on the original table shown at the beginning of the presentation. So driving forward, it offers kind of a fast and cost-effective way of doing that if you want to look at the entire genome and figure out these domains on a given individual. And note down with the molecular pileup at the bottom that in this domain, almost all of the genome maps are in concordant agreement, and it's pretty easy to see visually. So something that's really nice about this is that you can validate by eye, and of course there's software for doing that as well. So the second example that we wanted to drive, and Ali is really going to focus on in the second half of the talk, are where did we benefit from using these 2 long-read technologies in a hybrid approach for this de novo assembly? And one huge example is in chromosome 1, which is a 577 kb or 577 kb region. Then on HD19 predicts 2 very large gaps shown in these gray-purple boxes. But then on the super scaffold of the merge of PacBio and BioNano Genomics, you can see on the blue track that we resolved that assembly, got rid of the 2 gaps, found an inversion, and basically took this in sequence and placed it once we resolved the gap. And the nice thing about this is that it's validated by HD38. So it's important to note we're not using only structural variants that are validated by HD38. There are ones that are novel to this assembly, but we're utilizing this as an example because it is in fact validated by another assembly. So it's just a good presentation example because it's hard to find other technologies to validate the novel ones. So you can see that it's in complete agreement with HD38, and then again the molecular pileup of the genome maps against this super scaffold looks very clean in terms of the digest that Todd had mentioned in the preamble. So the reads are pretty clear in terms of resolving the structural variants there. So if you were doing some haplotyping, you would be able to see 2 populations in the genome maps themselves. So with that, I'm going to hand it off to Ali, who's going to describe basically the assembly pipeline and some of the other statistics and comparative analyses moving forward.
Thanks, Bobby, for going over all that. So what I'm showing you guys here is an overview of the types of analyses that we're really trying to target here. And you can really split this up on the left-hand side into de novo analyses and reference-based analyses. And one thing I really want to point out is what we're going to be focusing on is the de novo analyses, especially when we combine the kind of long-read data sets with the BioNano molecules. But the BioNano molecules can most certainly, as Todd had shown earlier and we've been kind of alluding to throughout, can definitely be used in reference-based approaches directly for structural variation detection and tandem repeat resolution, as we kind of showed in some of these earlier slides just a little bit ago. But that's really not what the focus of what we're talking about today is. What we're really focused on is these kind of de novo analyses and really trying to say, well, how can we extend some of these kind of conventional de novo approaches by combining the BioNano molecules. So just to go over kind of the rough kind of schematic, We start off with raw data sets from PacBio long reads and BioNano molecules. For the PacBio pipeline, we perform standard error correction approaches using the methods from Chin et al. Discussed in 2013 in the Nature Methods paper. And we develop an error-corrected read set. So this is a custom pipeline that we developed in order to handle human-sized genome sets. We then run long-read assembly pipelines. So this is completely independent at this point from the BioNano molecules. So the long-read assemblies consist of the updated version of the Celera assembly pipeline. This is developed by Sergey Koran and Adam Filippi's group, as well as a modification of Jason Chin's string graph assembly pipeline. So it seems a little bit convoluted to use multiple assembly algorithms, but I'll come back to this later on why that ends up being useful to us, and how that can be useful further on for kind of for diploid assembly applications. The output of this is assembled contigs that are going to be passed towards scaffolding. The BioNano molecules follow the kind of standard BioNano genome map construction approach, and the two are symmetrically scaffolded. So that's at this point right here, we have the two molecules, and rather than just using the genome maps to scaffold the assembled contigs, we symmetrically scaffold the two, and this is where we get kind of substantial improvements over what you would traditionally see. The result is now our scaffolded contig set. So just to give you kind of a high-level overview of what this leads to, I want to show you how the different data sets kind of build on one another. So this is a complete overview of all the different chromosomes all the different assembled chromosomes. So on the top line in pink, you're seeing different shades corresponding to the PacBio assembly. So as you go from light to dark here, that is indication of the PacBio contiguity. And the reason we do that is the PacBio assembly is actually a fairly complete assembly. There's not large gaps in the assembly. And in fact, sometimes the contigs overlap each other, as you would expect through kind of overlay out consensus algorithms. But the assembly contiguity is much, much lower than either the BioNano maps or the kind of the scaffolds. So the color intensity indicates the contiguity of the assembly in different regions. So below that, in dark blue, corresponds to the BioNano genome map de novo assembly. And below that, in red, corresponds to our first scaffold assembly, and this is primarily done with the Celera assembled contigs. And then the one in yellow corresponds to an additional iteration where we used a more aggressive assembly algorithm based on kind of these string graph approaches. And then below that is finally the chromosome ideogram. Lastly, I just want to point out that this cyan color corresponds to conflicts in the two assemblies. So for example, a case where the sequence-based assembly conflicted with the genome map. And we took a very conservative approach, at least we're taking that right now, in this kind of first attempt at kind of looking at what we can do with the data, and we threw out all conflicting regions in the assembly. So that didn't actually make it forward to the downstream scaffold. So if the sequence assembly had a strong disagreement with the BioNano genome map, in this case a strong disagreement was indicated by more than 10 mismap markers, we ended up throwing away that particular contig. In many cases, this is overly aggressive because of, largely in the case of the sequence assembly, you'll get errors at the end of the contigs, but also we have just different structural variations, which we'll come back to again at the end. The gray areas correspond to large gaps in the reference, and I point that out for several reasons. One, these are regions that Bobby had mentioned at the beginning are currently unknown. Oftentimes these correspond to centromere and heterochromatic regions. But this also limits our ability to place some contigs. When you have many megabases of ends, obviously you're not going to be able to map, and we can't really indicate where these contigs are going. So it's one thing that we've noticed. We're often able to push into these regions quite a bit, but there's not really a good map. It's very challenging to map in these regions because we don't know what's actually in the reference. As you can see, moving forward, so if we look at an example in chromosome 2 where my mouse is, you'll see that though there's a substantial improvement with the genome map from the original PacBio assembly, the movement in the V2 scaffold is quite substantial. So for example, in chromosome 2 here, we almost resolved the entire arm of the chromosome. As you can see, it was even a pretty fragmented genome map going in. Similarly, on chromosome 18, we had multiple genome maps on both arms, and we resolved both the P and Q arms completely. And they're entirely consistent with the underlying reference. So obviously, internally, there might be small structural variations, but the end and the beginning of these alignments are completely consistent. And internally, as we'll show in a little bit, you can see that the genome map alignments are really quite consistent with the underlying assembly. So what is the approach that we use to do this? Well, as I mentioned earlier, you start off with your NGS contig set. In our case, it was PacBio, and we do an in silico digestion using the particular restriction enzyme site that was going to be used in the BioNano genome map construction. The genome maps are passed in as, for people familiar with the technology, as they normally would. So you have these two CMAP constructions. You now align these two CMAPs to each other the same way you would align a genome map back to a reference. You just align them symmetrically to one another. Now, once you perform some filtering based on quality, you want to try to get the top alignments and unambiguous alignments between the genome maps and the NGS, obviously, to get kind of high-quality scaffolds. We then end up returning 3 different sets of outputs. One is you have leftover NGS contigs. So these are contigs that could not map to the BioNano genome maps. Largely, these leftover contigs are due when the contigs are too small. If there is only 50 kb of sequence, you oftentimes don't have enough NIC sites in order to accurately map the sequence. So that's one of the largest occurrences for the genome maps, but it also could be additional structural variants that didn't map at high quality, or just sequence that you didn't see in the genome map for whatever reason. You also have the merged hybrid scaffold. So these are maps that align to each other that we were able to join and form into a single hybrid scaffold, and this can consist of multiple NGS onto one genome map or vice versa, or chains of genome maps and NGS kind of symmetrically scaffolding one another. And then lastly, we have leftover BioNano maps where we weren't able to align any contigs to. We then iterate on this at a decreasing level of stringency. So we really want to get our highest quality placements first and then iterate through it. The reason we do this iterative procedure is to try to best place, to the best of our abilities, things like segmental duplications, and as such, we want to get the ones of the most highest confidence placed first. The resulting output after several iterations on this is, before I go into that, at each iteration step, there's one last position where we mark contigs as being inconsistent or consistent and eliminate them from further scaffolding. So in our particular case, the manual creation here was actually a fairly simple step. We just set a hard cutoff, more or less, which was if there was greater than 10, I believe, unaligned NIC sites, we considered it to be inconsistent, but if it was the top alignment, basically an unambiguous alignment with more than 10 inconsistent sites, and that was eliminated from analysis. So there's a pretty conservative approach, but we believe that this leads to less misassemblies, and which is why we went to it in our kind of initial pass. The result of this ends up being a FASTA sequence, which is formed by linking together the initial FASTA files, and there's a simple tool I wrote to basically use the predicted distance from the genome map of linked contigs, and then perform a simple seeded-based overlap alignment at the ends to try to merge the two FASTA contigs at that junction. And an AGP file, for those familiar with who've done kind of these scaffolds, an AGP file is essentially a scaffold of your sequence contigs based on whatever your particular scaffold is. So what are the actual stats on these assemblies? So we started off with a pretty reasonable sequence assembly using fairly aggressive parameters. We got to about 900 kb, but a large number of contigs and a fairly reasonable genome size. With the BioNano genome maps, we had a much larger N50 of 4.6 megabases. But what I want to point out is the substantial improvement that you get with the hybrid scaffold. So after multiple iterations, you get down to 31.2 megabases and 50 for the scaffold. So it's different than the full data set size because the scaffold is losing some genomic material. So this is an important thing to point out. The assembly is not completely representative of the scaffold doesn't completely capture everything in the genome. I'll go back to this later on when I compare to some other assemblies, but this is actually not uncommon with scaffolded assemblies. So if you compare it to other high-quality scaffolds, about 91% of the genome maintained that is actually pretty good quality. So we're pretty pleased with that. You get a substantial reduction in the number of contigs, and overall, the mean contig length is quite a bit better as well. Again, we know which contigs were scaffolded. Remember, we have leftover sequence contigs that didn't get placed within the scaffold, so we can always go back to those and look at those again. And as you can see, when you add those back in, the N50 goes down a little bit, and you get many, many more contigs coming in because most of these are very small contigs that couldn't get placed. So that's why the assembly size increases here. So what you're representing here is contigs that couldn't be placed, so you're sometimes double counting regions of the genome. And you also have structural variants, so that's intrinsically double counting regions of the genome. So these are kind of diploid breaks in the genome. So why is there such a dramatic improvement? So you see this from 1 megabase to 30 megabases, even though the genome maps are only 4 megabases. That seems kind of non-intuitive. Why are we getting such a substantial improvement? Well, it's because that the reasons why the contigs are breaking are somewhat orthogonal. So in general, I mentioned, or Bobby mentioned that there was a particular read-length distribution for PacBio. In our data set, recall this is somewhat earlier data, but most of our data is less than 10 kb. So for very large repeats, which, I mean, for most part, when you're looking at the human genome here, you're talking about small segmental duplications here or very large tandem repeats. We're not going to be able to resolve those guys. However, the genome maps are hundreds of kb inside, so most of them, except for very large segmental duplications, are not going to be broken on repeats. However, as you can see on the right, what does happen with the BioNano genome maps, at least in our iteration, as we only used a single nicking enzyme, is that you have fragile sites where two proximal nicking enzymes on opposite strands could lead to contig breakages. So we check that by looking at the distance of fragile sites throughout the genome. The mean distance is actually roughly RN50, but when you look at near our sites, they're greatly enriched. So you can notice the scales are actually changing to 1/20th the size, and the mean distance is going down to 0.2 megabases. So that's really what we're seeing. But that's actually extremely useful to us because it's orthogonal to what's happening in PacBio. We just get this huge improvement in our scaffold M50s. All right, so we did that very high-level overview of some of these assemblies. If we zoom in, how do they actually look when you align them genome nick site to nick site? And what you can see is that these are extremely highly conserved. So it's hard to see it on this particular image, I realize, but the fact that it's all gray means that basically every single one of those nick sites is aligned to each other. So it's a very, very high quality alignment of these regions. In some cases, you'll notice in this first one, you have a little bit of an expansion. That suggests there's probably some structural variations in there. But in general, you see some very high quality alignment of our particular contigs to the genome, even though occasionally, as you can see by the area I'm marking with my arrow, you do have regions which we did not cover. Well, one of the things I would say is even in regions in the reference that are gaps, there's a lot of regions marked by these kind of pink gaps. These are regions in the reference which are basically just blocks of ends. Even in these regions, we're able to cover a number of them, especially if they're small. We just run right through them. In some cases, our maps will overlap. So this is actually fairly infrequent that we see this occur because of the way we built the scaffolds. Most of the kind of orthogonal, most of the additional haplotypes are coming out separately in the sequence contigs that are separate from the scaffold as kind of additional contigs. But occasionally you get these things overlapping, and what we see here is these are very likely just alternative haplotypes for structural variations. And when there's very large gaps, there's usually a reason why the reference has a very large gap in that region, either through low complexity, alpha satellites, etc. But another thing is, even if we were to resolve that region, there's no way for us to see it by this image. So I just want to point out that these large gaps that you're seeing here, they're just ends in the genome, so we can't map anything to those. So not to draw alarm to anything. And in some cases, as you can see where my arrow is up here, we do go into the actual gap regions. Well, of course, when we're looking at assembly, one very useful thing to do is to compare it to other assemblies that have been out there. So one of the kind of, I'd say, exemplar papers in this space has been from David Jaffe's group, who did an NA12878 assembly using Illumina data in 2010. So this data set used a number of kind of high-quality paired end and phosmid libraries. So you have standard kind of short reads with 155 base pairs, but you also have jumping libraries of 2.5 kb and a phosmid library of 35 kb. And all of these were used at very high physical coverage. So you have a very high physical coverage assembly. This is, in my opinion, one of the best available assemblies for this particular genome, at least that I was able to find, and very high quality. Perhaps the gold standard right now in that particular space. So when you compare this assembly, you can see that it has a very, very high N50, so that is 11.5 N50, and one of the things that came out from this paper is just how 11.5 megabase in 50 is how much better this was than the other available kind of short read assemblies at the time, when you have a SOAP assembly, which is just several hundred KB. So this is a very high quality assembly. And it's almost on par, in fact, with the original QREP assembly, which is 17.6 megabases. So how does our assembly compare? So we start off with those PacBio Contigs, as I mentioned, which is about an 896 Contig at 50. So that's better than any of the Contig and 50s that have been out there to date, even with the Sanger sequenced assembly. So we're several hundred KB better than that, and that is really, really helpful for anchoring these BioNano Contigs. And then when we actually look at our hybrid scaffold, we're at 31.2 megabases, which is better than any of these other assemblies, but quite a bit. And one thing I want to point out is this 11.5 megabases, this covered also 91% of the genome, so we're almost one-to-one on what we're covering on the genome. So it's not that they covered way more of the genome than we did. We're really, I'd say this is not quite an apples-to-apples comparison because of the short read sequence quality, which I'll get into at the very end, but it's about as close as I think we can get with current technologies. And this used, again, cloning libraries with plasmids. So we feel reasonably good about that, but one of the things that, of course, everyone's concerned with is it's not just how contiguous your assembly is or how good your M50 is, but how reasonable is the kind of assembly quality. And again, I'm going to focus on contiguity of the libraries as opposed to the assembly quality. So one thing I will admit is that our sequence level quality is lower than these short read assemblies, and that's something we're kind of continuously working on. And it's quite a bit lower. In our case, you know, there's millions of indels that we miss. So for this, our V2 scaffold is shown at the top, and the all pathology is shown below it. What we're looking at here are how many breakpoints do we see relative to HG19, i.e., when we align our assemblies to HG19, how many times is our alignment broken where we have more than 5 unaligned nick sites? This could be at the end of a contig or internal to a contig. How many times do we have greater than 5 unaligned nick sites in a particular interval? And what we're seeing here is that in our particular assembly, we have 39 intrachromosomal, which are likely, though of course you can't guarantee it, structural variations, and 7 interchromosomal, which are almost certainly misassemblies. This actually compares fairly favorably with the all-paths assembly. Keep in mind the all-paths has more contigs, more scaffolds, so there's going to be potentially more events, but in general, it's comforting to us that we're kind of on par or slightly better than what they're showing there. And the other point to bring up is, well, what about all the unique bridges that we make versus the unique bridges that all paths make? Well, we also find that we also have 248 unique regions that we bridged that weren't bridged in the other assembly that seem to be consistent with HG19. So we're not just having less wrong events, but the additional events we do predict tend to be right. It's interesting that there's a large number of events that they predict that we don't. So it suggests that there's a lot of things that they're doing that we could really learn from, and that there's still a lot of use in having these clone libraries, because there's things that obviously they're able to resolve that we aren't. So lastly, we want to look at the conflicts between the two assemblies. And so when you look at the conflicts, so these are regions that all paths and our V2 assembly disagreed with each other when you align the two contig sets to each other. So there's 2 of these where the V2 was clearly inconsistent. So all paths was right and we were clearly wrong. There's 28 where the all paths were clearly inconsistent. And there's 10 in which both assemblies were potentially consistent. And so this is kind of interesting, or they map to different locations. But this is potentially cases of structural variation where we're finding 2 different haplotypes. So here's just an example of one of those. And so we can look at this on an all paths scaffold versus versus our scaffold. And you can see at the end here, marked kind of by that red point, you see a region that we don't align between the 2 scaffolds. So if we look at that region and then try to track it when we take the reference to back to all paths and the reference back to ourselves, what we can see here is that that region actually aligns in both. So there's clearly a strong difference at the edge in the all paths assembly, but likely, based on how the rest of the genome, it could be just a structural variant, or it could be degradation in the assembly. It's somewhat hard to say, right? But it's not clearly that one assembly was right or wrong. So this is one of the 10 ambiguous cases we flagged. But when we look at our assembly in that region, it's extremely consistent with HG19, so it tells us that we believe we're on the right track there. So another example is where we think there's a potential discrepancy on their end is here again is an area where the contig doesn't align. And you can see that there's actually quite a few kind of internal discrepancies. So if we look at that region, in the 3 assemblies, we can see that our assembly is still completely consistent from end to end with HG19. In the all-path assembly, in that particular interval, it looks like there might be a structural variance, so it's at the end of this box. You can see there's a gap here which probably led to this discrepancy, and this could be a structural variation again, because it picks up quite nicely after that, but then the rest of the contig seems to misalign to the genome, indicating a potential, potential kind of suspect region on the genome. Of course, we're not always right either. So if we look at just regions that we had discrepancies in the genome, so I mentioned that there was 39 of these intrachromosomal events. So what are these intrachromosomal events? Not all of them are clearly structural variation, though a lot of them are. Here's one that's particularly confusing. So you'll see, if you look at HG19 relative to our assembly, we see 3 distinct events. And this also spans over a large number of gaps in HG19. So we want to say, well, HG19 is imperfect, HG38 is a newer assembly, let's take a look at that. So we look at HG38, which just came out a few months ago actually, and we can see that we have a much more consistent assembly with HG38, but there's still what looks like a rearrangement. So we tried to examine single molecules in this junction, and what it turns out is there is a segmental duplication right at this junction, and it's basically impossible for us to say what is the correct order in this region based on our molecule. So again, this could be a misassembly, but again, it could be that there's just not information. It could be that HG38 is still incorrect here. We're obviously not going to say one way or the other based on the information that we currently have. In general, though, we see that the gaps tend to be resolved, these gap regions, and we also have a decrease in overall rearrangements when we move from HG19 to HG38. Here's another event below where we have 2 events suggested by this inversion. We're again closer with HG38, but it is still a discrepancy. Again, this discrepancy at the junctions is mediated by a segmental duplication. So just to finish up, I realize I'm kind of a little bit over time, but we're continuously trying to improve the accuracy of these assemblies. So we're trying to work on that. The biggest area is in the long read assembly. It's still less accurate at low coverage. Again, we're at fairly low coverage for typical long read assemblies than a short read assembly, and we're trying to see if we can use the short reads to really improve some of that. And we really would want to use kind of maybe a 3-tier hybrid sequencing strategy. We'd also like to try to circumvent some of these desert and fragile sites in the DNA nick sites. Potentially, it could even improve the scaffolding sites further. And so, obviously, I know people have been doing a lot of different things at BioNano with combined multiple nicking enzymes, things like that. I think this is a temporary side point, but worth mentioning. We also want to reduce the requirement for at least 100 kb contigs for accurate mapping. So in our case, we are really, when we actually look at it, we're almost throwing away all our contigs less than 100 kb in terms of scaffolding. And we'd really like to be able to recover some of those if possible. So in the sense of after we've done all the kind of scaffolding, the regions of the genome that are left are actually quite small. We can probably eliminate such a stringent requirement. And really look at the sequence assembly graph, which is what we've been doing now to kind of reconstruct local haplotypes in the string graph to say which are tolerated paths in the genome map. One thing we didn't do systematically was examine TRs, but clearly, as Bobby showed, they're very powerful, and we can really resolve. We have a couple interesting cases that we resolved exclusively with the BioNano raw molecules. And I think this is really an exciting area because these are the regions that no one's really looking at in the genome. And lastly, the way we are doing this currently, the genome maps are constructed tend to compress haplotypes when the haplotypes are somewhat complex. So when we do that, we have to go back to the sequence data, and then we go back and use the long reads and the short reads to do haplotype phasing. So what we do is we remap all the short reads to the assemblies, we call SNPs, we then use the long reads to do readback phasing on this data. Ideally, and this is something that we're trying to do now, and I know that this is something that BioNano is actively working on, is we'd like to be able to get a diploid construction of the genome maps put in, as opposed to doing the diploid analysis after the fact. So we do actually get reasonable contiguity of the diploid assembly, and in fact, our phasing, when we go back on this, is over 99.9% consistent with the, or sorry, is either over 99% consistent with the trio-based information we have for this family. So it works reasonably well when you have a good trio and you have these information present, but I think at a structural level, there's a tremendous amount of growth we can have by really incorporating the sequence graph with the genome maps directly. I'm just going to end here in acknowledgements. There's a lot of people to thank who kind of worked on the PacBio side, a number of collaborators at PacBio, Weill Cornell, Cold Spring Harbor, and Rockefeller, And of course, with the BioNano, with all the folks at BioNano, I know Andy Peng did a lion's share of the work there in the end, but Heng Dai also contributed a tremendous amount, as well as UCSF's Pui Kok for giving us a lot of this data to work with in the first place, and a number of folks at Mount Sinai. And I think, yeah, we're open to any questions that any of you may have. Thank you.
Terrific. Thank you, Bobby. Thank you, Ali. We are now in the Q&A portion of the webinar. The floor is open for questions. You can type your questions into the chat box, and we will read them out. We already have several questions in. So the first one for Bobby and Ali is, can you comment on the usage of this combination of technology methods in resolving complex variation structure, particularly in cancer genomes?
So that's something that, so certainly it's something that we've been very interested in, but the issue that we currently have is oftentimes, as I'm sure the person who asked the question probably realizes, the heterogeneity is the question of, if you can get a pure enough sample to do it, then there's something interesting to do. But in our case, one of the issues is you have to sequence to such great depth on the long reads that the long reads, it tends to just not be worth it. It's just not cost effective.
So what's going to drive some of that, Todd, is basically, as single-cell isolation technologies move forward and we can finally move beyond just using WGA, going that avenue will help a lot. But right now, as Ali is saying, the depth of coverage required would be so high to deconvolve, the heterogeneity in the sample that it's a little tougher for somatic samples.
Right. And again, one of the things is, on a genome, we can do reasonably well. We're kind of at the minimum, I would say, of coverage, what we're using right now. So on an impure sample, having to do several 100x coverage is just not financially tractable, we think, right now, or at least for us.
Right.
So thanks, guys. And when you're talking about these coverage depth requirements, are you referring to the PacBio?
Yeah, so we're basically referring to, the question was specifically asking about the hybrid approach. If you were just using genome maps, I mean, you can generate higher depth much faster and cost effectively. And I guess you could probably address that component if you want, Todd.
Yeah, happy to, and my colleagues here in the room can pitch in too, but we actually have many customers now that are buying the IRIS platform specifically for cancer research, given the implication of structural variation in cancer. So it's early days. We have done some work already with cancer cell lines and gotten very promising results. And in some cases, we've been able to show that we corroborate next-gen sequencing results, and sometimes we show errors made and can uncover errors made in the next-gen sequencing. So it's highly complementary to those technologies. We are now moving into other types of samples. We're running, New York Genome Center is running their first biopsy samples, and we're looking into heterogeneous tissues, and so that is definitely on our roadmap and of keen interest to us here at BioNano to start exploring the cancer work. It's likely, to your point, it's likely that higher coverage depth will be required, but we have yet to really determine what that coverage requirements are. Anybody want to comment?
This is Han, Chief Scientific Officer by Nano. I want to make one simple comment. In the cancer, there's a lot of heterogeneity, and pay attention, these two methods actually are single molecule platforms. So that's a very unique combination as well. So in this case, BioNano has very long contiguous molecules. Sometimes, as Ali showed earlier, there's certain area has some kind of overlapping. He pointed out could be heterogeneous haplotypes. And in this case, in BioNano, because every molecule is recorded, actually, you can go back to the raw molecule data, you're able to see a fraction or the 50% segregation of those molecules there. So there's a lot of information there. So that's why I want to show this, the value of it to map further into the subpopulation heterogeneity of a cancer sample.
And I will say that, so we definitely, for structural variation applications, we definitely do that. I don't want to indicate that we don't do that with kind of long reads, just as people do it with short reads. We definitely do that. I was specifically talking about the way we did the hybrid scaffolding. We do a bunch of assumptions about coverage and symmetric coverage applications to make things work, and that's why I think that might not be trackable. Certainly for structural variation, the long reads work just as well as the short reads, and we've seen that in terms of seeing kind of like local rearrangements. So I don't want to, yeah.
Great.
Okay, terrific. I'm going to move on to the next question. This is aimed at Ali. Could you comment generally on the scale of improvement introducing additional or multiple nicking endonucleases that they would present?
So we haven't seen it on this particular assembly, but we did use it on another assembly.
Yeah. So I can actually comment on that on the molecular level, Todd. So we did utilize that on Plasmodium assembly, and by doing 2 different nicking approaches, you can both validate some of the calls you make between the 2 approaches, but also use the data to your advantage to definitely reduce the number of scaffolding contigs. So as you might imagine, by introducing other nicking enzymes and data that way, you can use it in both ways to improve the assembly. So that's definitely a good thing if you have low complexity assemblies or for structural variants. I think if you were having a relatively diverse genome that was small, like a microbe or something, it's not really going to help you out there, and you don't need it. It's not required. But for these more complex situations, it is.
Yeah, so I don't think that we would get just in the so on the Plasmodium assembly, it actually made a huge difference. It basically resolved the entire thing.
Which makes sense because of the AT-rich.
But I don't suspect actually we get as huge a difference in the human because when we actually start to look at where we're breaking, there's just so many cases that are these segmental duplications.
That's right.
That and we're just saying centromeres. So imagine we have maybe 200 contigs, right? You imagine that you're going to get 40-something, just 40, 45, whatever, for just based on looking at the way the genome is, right? That's out there. So the rest of them are just there's going to be these 100 kb segmental duplications. So I do think it'll improve it. I don't think we're going to get a doubling N50 or anything like that. I think we're pretty much the N50 improvements at this point are going to be pretty small.
But for instance, if anybody out there is working on large plant genomes, for instance, it may help there because of the low complexity of those genomes.
This is Han BioNano side. I want to make additional comments. So the value of this BioNano single molecule because it's intact contiguity, we do have evidence to show there's no upper limit to how large we can make those channels. So there actually is really limited by sample prep. And recently we actually show a single molecule span 1.75 megabase. The entire molecule are composed of 2 type of SVs. So this SV can go on and on for megabases. So in this case, when you have this extremely long molecules, you really reduce these ambiguities. And so I think that's the value comes in.
And reduce the need for multiple nicking enzymes is the point?
Well, I specifically address the depths.
Yeah. Back to the
certain point is you just increase depths cannot span those things. It's sometimes a single molecule very long can tell more story than a lot of depths increase, right?
I will also add further comment that the IRIS instrument does come with 3 lasers already on board, and we are working to get a 2-color assay up and running, hopefully by end of this year, which will allow you to do multiple nicking enzymes at the same time rather than subsequently with the single channel. So we definitely have an eye to increasing nicking enzymes and even other labeling techniques that we're developing here. The next question is, how do you determine if a structural variant is homozygous or heterozygous?
So Bobby was just saying that the way we do that is we do it through pile-up analysis. For the short reads on the PacBio side, what we do is we basically we cluster together the reads on each side, and we say, okay, well, here's a particular structural variant that we predict. And then are there reads that do not agree with that? So it's basically just align those reads back to the particular structural variant. I actually added in one last check, which I didn't include here, which is because we have the short read data and the reads are so long, we do a second check, which is we look for proximal SNCs upstream and downstream of the predicted structural variant, and we look to see if the partition that we predict, so we're predicting that some reads are on one allele, some reads are on another allele, do those separate out into 2 distinct haplotypes by SNP. If they do, then we have high confidence that is in fact, heterozygous event. Otherwise, there's ambiguity. So It's both by looking for these reads, but then also validating by SNPs. Because the reads are, several, the average SNP distance on this genome may be, several kb, but we'll usually have at least a few reads that are in the 6 kb plus range, and so we typically hit at least a couple SNPs in that process. On the genome map side, right now, the way we're doing it is by looking at molecule pileups, like the ones that Bobby showed, and we're using that to kind of validate large structural events.
Terrific. Thank you. The next question is, have you tried any wet experiments to validate the structural variant results?
Yeah, so we've done some targeted PacBio sequencing, and obviously we could run some of the longer sort of long-range PCR or Sanger-type experiments for validation. So we didn't end up including that that I know of. Ali can probably comment if...
So one of the issues so we can do that for the PacBio, but the genome maps, a lot of the events are just too large for us to validate. So instead what we did is we took an orthogonal data set, in this case the short read data for the events which were tandem repeats, and we looked at the coverage distribution of those short reads. And based on that, we used to kind of validate empirically that way. But we don't have a good way. So we're actually, a person in my group is actually trying to develop a way to actually do this right now. But it's been really one of the big issues we have is if you predict a 100, and so in this case sometimes they're 377 kb event, we don't believe that validating the junction is necessarily sufficient. We really want to kind of span in through an unambiguous region, and we simply can't do that with PCR, which is the way we've traditionally been doing it.
So
for copy number changes, it's a little bit easier, but for those copy neutral events, it's really quite challenging.
Yeah, you have to get pretty creative with your technology because to Ali's point, you're not going to get beyond about 20 kb with long-range amplification methods. And you could start doing the breakpoint analysis using like mate pair plus PacBio, but it turns into an assembly problem of its own to do that.
So to be honest, I even looked in the NA12878 plasmid jumping libraries, and many of the events we're showing, the junction is even bigger than what you can see in the plasmid jumping libraries.
Right.
So we would need to do something like a back jumping library.
Or bigger.
And it's just, honestly, it's largely just a cost issue of constructing those back libraries.
Great. The next question is, will the introduction of BioNano molecule maps or genome maps into this three-pronged hybrid strategy effectively reduce project costs by way of decreasing PacBio, for instance, physical coverage targets in your design of experiments?
So, I think that that's likely true to reduce the cost. I think the main thing is you're going to have. I want to not mislead. You're not going to be able to drop the coverage of the PacBio data down to 10x because of the way the error correction works.
Right.
You need to have a reasonable starting point, and I think that what we have here is kind of the absolute minimum.
Yeah, so we've found from other genomes you probably want to work in the 30 to 40x coverage regime as your minimum, so we're already pushing that limit. What you can say towards the can you drop the coverage of PacBio is just that, as their QV improves and read length improves there, you then may be able to start doing that. So it's going to be kind of you tracking the improvements on the single molecule, single base sequencing on PacBio side versus the length that you're getting from BioNano. But using BioNano in general definitely doesn't mean that you then have to go to 100x on the PacBio side. Which is improving in the assembly contiguity in general because of the gaps that you're filling in and getting across structural variants.
Right. I mean, and certainly, but there is advantages in the sense of like if you go higher coverage, then you can get higher accuracy. But yeah, certainly you don't have to push it up to 200x to get those really long reads and those 100x. But I think one of the things that we're suggesting here also is it's not just that you're decreasing your PacBio costs. It's also that you're getting kind of just really, really long stuff that you wouldn't see necessarily otherwise.
That's right.
So I think that both technologies have tremendous advantages, and I think that the way we did it is currently one of the best. We believe, obviously, we wouldn't have done it this way. We didn't think it was one of the better ways of doing it because you really get that minimal contig size that, as you can see from those kind of other short read approaches, you're really getting very small contig size, even if you have high scaffold sizes. And those small contig sizes don't do as well when combined with the genome maps. You really want to have kind of these slightly larger contig sizes. But yes, we could probably get away with having a 500 kb and 50 and really getting pretty similar results as opposed to a megabase or as opposed to 5 megabases.
And I totally concur with Bobby and Ali. I want to quickly add is the combined analysis time is shrinked. I mean, that's the very important thing, because you're reducing the cost by you reach confidence of a certain area much quicker. So overall, you actually reduce the time your analysis. That's actually a lot of cost. And also certain areas, Bobby mentioned, that you cannot even touch before. Now you're able to see it.
Yeah, that's a really good point that we completely agree on as well, because that is a huge time sink for these larger diploid assemblies.
Right. You might be tying up your machine for quite a while.
Yeah. Okay, I think we're gonna end it here. I want to thank our speakers, Bobby and Ali. Thank you very much for a great talk and the question and answer session. I'll remind our listeners that this is the recording of this webinar will be available on our website within the next day or two. And please keep an eye out for our next webinar in our series. And if you aren't a part of our BioNano community, please sign up. Go to our website and you can do that there. Thanks for tuning in, and we'll talk to you next time.
Great.
Bye-bye.
Thanks, guys.